Okay, so this is going to be an extension of the things we talked about last time, which is non-allelic homologous recombination, which is to say that recombination, of course, involves sequences that are similar or identical to one another, and they are homologous because of that fact. Allelic means that the sequences occur at equivalent positions on homologous chromosomes. So these are, of course, very similar to one another, and recombination is a normal process that does not cause any problems usually. Okay, the problem arises when we have so many repeats in our genome, which are similar to one another in sequence, but are not allelic to each other. So such recombination between such sequences can occur and can have very bad effects on our genome, on our genes. So this is also called illegitimate recombination because the sequences are similar, but they're not actually allelic, even though the sequences are sufficiently similar to allow homologous recombination. So just to illustrate the fact that this can be dangerous to our genome, here's a table of some diseases that have resulted from this sort of recombination, mostly involving Alu repeats, but also some involving L1. So not to think that all these diseases are always caused by this type of recombination. For example, breast cancer causing mutations in BRCA1, there's many different mutations in that gene that can lead to a predisposition to breast cancer, and only this one that we're talking about has to do with Alu. But nonetheless, there are many examples where this type of recombination has led to defects in genes, ultimately to the creation of genetic disease. Okay, so now I'd like to talk a little bit about the endogenous retroviruses because they do cause some issues with our genome. Okay, so here's HIV, a real retrovirus. It is represented in its DNA form as it exists in the chromosome when it would be sitting in an infected cell getting ready to produce new virions. Okay, now this diagram is very representative of basically all retroviruses, of this type at least, in the way the genome is organized. On the very left, we have the LTR, long terminal repeat, and there's another copy of that exact same sequence on the right. So that's the characteristic of this type of transposon for sure. So they all have that, and they all also have a genomic region that's divided up into basically three parts called GAG, POL, and ENV. This is a very characteristic genome organization. This is HIV, and it does have additional regulatory proteins, VIF, VPR, etc., in addition to those standard general retroviral genome organization. But this is very typical of the genome. Okay, so the GAG, POL, ENV organization, flanked by the long terminal repeats, is very typical. But the virion, of course, is going to contain RNA, not DNA, and it will get reverse transcribed in the process of infecting new cells. Okay, so there are a lot of retroviruses that are very much like this, including, just for example, mouse mammary tumor virus, avian leukosis virus, human T lymphotrophic virus, etc. So these are infectious. They produce infectious particles that are transmitted from cell to cell and from individual to individual, as viruses do. Okay, another general feature is that the LTR, left LTR, contains a promoter. So this is the only promoter that's present in these viruses, and so they use this one promoter to produce the RNAs that will make not only the new copies of the viral genome for incorporation in infectious particles, but for their own RNAs to produce protein. So we're going to have the left LTR as an important feature of these viruses. Okay, so retroviral protein production is really weird. The weird fact is that the retroviral proteins are translated initially as what are called polyproteins. So when we talk about the GAG, POL, and ENV, those are the initial proteins that are translated, but they have to be proteolytically digested, processed, to form the final proteins that are going to be actually part of the virion. Okay, so the protease that's responsible for this is encoded by the retrovirus as part of the POL region of the genome. Okay, so the GAG region is for capsid, matrix, membrane glycoproteins are from ENV. Okay, so what this looks like is if we consider the GAG region that's present in the longer of the full-length RNA, that will be translated to form a protein, which is the precursor to the eventual core structural proteins, the P18, 24, and 15. So that is one translation product which counts as a polyprotein because it will be ultimately producing 3 different proteins from that single initial protein. Same for the Pol region, that is going to produce a precursor, P160, which will then be proteolytically cleaved to form the protease itself, but also the reverse transcriptase and the integrase, which are of course crucial parts of the viral life cycle to get it to go and insert in a new location in the genome or some other genome. Finally, the M region produces by a precursor, which is the M precursor, which is then proteolytically cleaved by the protease to form the GP120 and GP40, which will end up being on the surface of the virus extravirally and are used for infectious purposes. Okay, so the GAG region becomes the capsid and nucleocapsid. Pol forms reverse transcriptase, integrase, and protease. N will form the surface proteins of the virion, and then the regulatory proteins are actually translated separately. So one important feature of these is that these proteases are very important for the virus, obviously, and that's why protease inhibitors have become really crucial anti-HIV drugs, because the virus needs to have this protease to produce its active reverse transcriptase, for example, in order to continue the life cycle. So these are very effective inhibitors. Okay, so what these are retroviruses which no longer go from cell to cell or from person to person or organism to organism, but rather viruses that have taken up residency in our genome and remain there. They don't transfer at all. They're mostly decayed in the sense of having a lot of mutations in them, most of them have little protein coding capacity, but some can be transcribed. Okay, most of them are not active at all, and the only reason you can tell that they're there is just looking at their sequence, comparing them to existing retroviruses. But they're not all completely dead. They can have functions in cells that we'll see. Okay, so in terms of classification, they are classified according to their ages and in what organisms particular ones are found. The oldest ones are the HERV-L group, which apparently colonized the mammalian genome very many years ago. Then we have the HERV-K class, which came later, the HERV-W class, and finally there's some more recent HERV-K insertions, which occurred approximately 670,000 years ago. So these have been around for a long time and are characteristic of mammalian genomes in general, but we have maybe more than a lot. Okay, so they're now a permanent part of our genome. They inserted somewhere in the distant past, and they are not active as viruses, but they're still there. They are inactive in the sense that there's no completely active element where all of the open reading frames are still intact. But there do exist examples of, for example, the reverse transcriptase and the N for sure, that are essentially intact. Within the genome, where we have 1,000 copies, there is at least one essentially active version of all of the viral proteins. Okay, there are some examples of complete ORFs, but no intact actual virus. Okay, so these are vertically transmitted, which is to say they're passed from parent to offspring, and they're not infectious. Okay, so here's the vertical transmission idea, comparing what happens to a normal virus versus what happens with these guys. Okay, so in a normal virus, you have a virus, and somebody gets infected with it in their somatic cells. There, the virus can replicate from one cell to another, passing itself within the body, but it can also be transmitted to another person. So this is the normal way in which viruses go about conducting their lifestyles. In contrast to this, our HERVs are transmitted vertically, which is to say from parent to child. That at some point in the distant past, they were inserted in the germline of the person, our predecessor, and have remained there. So they're there in our genome, and they're passed down like any other Mendelian type DNA sequence. So one doesn't get these from the outside or from anybody else. They're passed to us from our parents. So this is to convince you these are real retroviruses. Here's an example, a different sort of diagram, but it shows that it has all the expected features of a retrovirus. It has a 5' LTR, or left, has a 3' LTR. It has a GAG, POL, M regions, just like HIV does. The protease is a little bit different, and it's translated from a slightly different way, but it's still right there in its customary location among retroviruses. So this certainly looks like a retrovirus, no question about that. Okay, so in terms of its interactions with our genome, there's so much of it, it must be doing something. Well, a lot of the copies are actually just LTRs, like they don't have the GAG-POL-N part, they just have the LTRs. So these are called solo LTRs, and so they have no protein coding capacity. On the other hand, they do contain the promoter, which would have driven the retroviral RNA expression, and so they can actually become activated some of the time. Now, mostly they're silenced with the usual sorts of, histone modifications that are silencing and 5-methylcytosine, etc. But they can actually become activated under some conditions, and they can, under some circumstances at least, influence the transcription of nearby genes, which can be of concern. Okay, so here's a particularly weird example of interaction between this retrovirus and our own genome, and that's the existence of these proteins called syncytins. Well, it turns out that since the N proteins are the source of these syncytin proteins that are actually used during formation of the placenta during early embryonic development. Okay, this is bizarre. Okay, so the retroviral N protein is what is used by the virus to bind to and fuse itself with a new host cell membrane. So these are considered fusogenic because they facilitate membrane fusion during the viral infection of a new host cell. So in retroviral infections, the M proteins, the surface and transmembrane proteins, do that exact function. They facilitate the fusion between the virion membrane and the cell surface. Well, it turns out that we have actually co-opted or X-adapted these proteins for use during our own embryonic development because these proteins are actually produced during early embryonic development, during which time they actually facilitate the fusion of cells to form the placenta. Now, this is totally bizarre. Okay, so looking at what these proteins look like and thinking about what they do, these are derived from the retrovirus. We've got the syncytin 1 has a surface part, the transmembrane part, the SU and the TM from the M region, and there's actually a proteolytic cleavage that occurs just as if it were within a retroviral particle itself. So these are retroviral proteins, really. So the connection here is that if you consider what the retrovirus is going to try to do when it's going to try to infect a new cell, it has its surface protein, which recognizes a receptor on the susceptible cell, binds to it, and that promotes fusion between the external part of the virus, which is a membrane, and the membrane of the new host cell. So this is a natural part of retroviral infection, whereby the capsid is then delivered to the host cell for the purpose of starting another infectious cycle. Well, that same thing happens during cell fusion, where you have the M protein sitting on the surface of a cell, recognizes a receptor on another cell, and in this case, the membrane fusion event leads to the fusion of the two cells, where you form a syncytium, which is, say, a cell that has more than one nucleus in it. And this type of cell fusion event occurs during formation of the placenta and is actually guided, at least in part, by these M proteins from our endogenous retroviruses, which is completely, utterly bizarre. So this membrane fusion thing has been used, and this is not the same virus, but other mammals have the same process. Now, this process of forming syncytia, which is to say cells that have been fused with each other, can be a problem, particularly if you want to create cancer cells, etc. If you've got these being expressed on the cell surfaces, you could have syncytium formation under conditions where you don't want actually to do that. Okay, so last time we talked about equal sister chromatid exchange, and I will reiterate a little bit about that this time. More importantly, though, we're going to talk about intrachromatid, which is to say exchange involves repeated elements which are within the same chromatid. And there's two ways in which that needs to be considered. One is when the repeats are in the direct orientation, which we'll see, and that leads to deletion. And the other form is intrachromatid exchange, which involves sequences that are inverted, which is to say pointing in the opposite direction. And those lead to inversion of the sequences between the repeats, with bad effects sometimes. So especially non-allelic homologous recombination involving transposable elements can occur because there's so many of them. It's kind of amazing that we don't have more of a problem with this than we do. Okay, now lots of times when you can see something like a LTR that's just sitting there in the middle of nowhere, you don't know how that arose, right? The example of intrachromatid versus sister chromatid exchange both could lead to that same outcome. So you don't always know what happened, but you know that something did to lead to the existence of a solo LTR. So I'm going to give a couple of examples where that's happened. Okay, so remind you what a retrotransposon looks like. It has a certain genomic region that encodes its protein coding genes, but it's flanked by LTR, which is long terminal repeats, one on each end. And there are many examples in our genome where we do not have full-length retrotransposons or endogenous retroviruses, but rather instances where just the LTR is just sitting there in the middle of the genome. Okay, so one way in which that can occur is, as we've seen before, with unequal sister chromatid exchange. So what happens is the two LTRs will line up, but out of register. Instead of the first one aligning with the first one, and the other end aligning with the other end, they line up such that the first one is aligned with the last one and undergoes recombination. So when that happens, you can have the formation of a fusion chromosome, which actually has two copies of the transposable element with an LTR in the middle, or a solo LTR. So to follow these recombination events, start on the left of the chromatid, go to the right. When the X occurs, go to the other chromatid, and then continue. And so you can see the outcome of these recombination events. And this is the same as we've seen before for sister chromatid exchange. Okay, the intrachromatid exchange mechanism is rather different. It will involve the two LTRs on the same chromatid. In the first example, where we have direct repeats, and that leads to deletion of the region between them. So it's not so easy to align these sequences because they have to occur in the right order, as you'll see. If it's ABC, we have to line them up as ABC. And that is going to involve twisting the molecule around to allow the homologous sequences to pair with each other. So we twist the molecules around, and then we do the recombination and look to see what the outcome is going to be. So here's one representation of this, where the little red things with the arrows are a directly repeated element, meaning the letters are ABC, ABC as before. Now to get those to pair up such that they're pointing in the same direction as each other, you've got to twist the chromatid around. But once you've done that so that the homologous sequences are aligned, you just do the crossover and see what happens. Okay, and what happens is basically deletion of the region between the repeats within the part that was involved in the crossover, and then a loss of that information from the chromosome itself. Another way of showing this type of inverted or type of twisting is here. This is essentially redundant with what I just said, that the little blue pieces are repeated sequences, the chromatid twisted around in order to allow recombination between them. And if you follow the recombination event, you will see that you have a little circle that's lost from the chromosome and a chromosome that's left with just the repeated sequence, one copy of it. Yet another example of the same thing. The figure on the left shows the arrows are the repeated sequence. A, B, C, and D are to show you what happens when you align and recombine those. So you align them, do the recombination, and you find out that you lose the region, including B and C, and you're left with one copy of the repeat, which includes A and D. All right, so recombination between LTRs can lead to the formation of solo LTRs. So I'm going to illustrate that here. Okay, so here I've shown A, B, and C representing the LTRs, and you can see they're in direct orientation because the one on the left is reading ABC, and the one on the right reads ABC. So we align our chromatids by our chromatid with itself by twisting it around, and then we make a recombination event within the LTR, between the repeated sequence. So what happens is, of course, you have a recombination event. Start with to the left, go through AB, cross over to C, and continue to go to the right. So you can see that you end up with a chromatid, which is now missing part of the information that between the two repeats and the region in between it. Okay, drawing that here, I'm following the recombination in yellow. The previous gene, AB, cross over to C, next gene, and that leaves the chromosome missing some of the information that it used to have, including one copy of the repeat. Now in pink, I'm showing the part that's deleted, and that was what's left over after the crossover event occurred. So this type of recombination leads to loss of information from the chromosome and a deletion of sequences, and is believed to be a major reason why we've got so many solo LTRs, which used to be part of endogenous retroviruses. So we've got a worksheet for practice, which is essentially the same, only we're using ALUs instead of LTRs, but the idea is exactly the same. We have a gene, which includes ALU elements within its introns, which is not exactly a rare situation, and we've got 3 exons. So we can actually put these ALUs through a recombination event, which will lead to the deletion of exon 2, and that's our question. One easy way to do this, of course, is by unequal sister chromatid exchange, where we're going to exchange between ALU, we're going to shift it over such that the ALU, which is between exon 1 and exon 2, is aligned with the other ALU, the one that's between exon 2 and exon 3. And if we have that recombination event, we're going to end up with a chromatid which has exon 1, ALU, exon 3, with exon 2 missing. Now the reciprocal event ends up being a duplication of exon 2, because if you follow that, you get exon 1, ALU, exon 2, and then the ALU recombination, and then you get another exon 2, ALU, and then exon 3. Now this is sister chromatid exchange, and that does not lead to deletion in the same way to a chromatid, but you still get the event where you're deleting exon 2 from one of your chromatids. So the other way to do that is to align the ALUs the way I've drawn in red. The little red things are the ALUs, and the arrows indicate their direction. And a crossover event between them you can follow with the green chromatid, starts in exon 1, and then crosses over. The looped out part is exon 2, and then you get to exon 3. And of course, the looped out part ends up separately as a circle and is lost. Okay, so the second type of event is when you have recombination between inverted repeats. And that's a different situation because you don't have to twist the chromatids around quite so extravagantly, but you do have to pair the repeats such that they're lined up ABC, ABC. And that recombination event will not lead to loss of information, but it will lead to an inversion of it, which can be devastating if it occurs in introns within genes. Okay, so this is an example of a real mutation that really does occur in human populations. The repeated sequence is called F8A, and you can see 2 of them in this factor VIII gene region. One is upstream of the promoter, and the other is within the gene itself, within an intron between exons 4 and 5. Notice that those are arranged in an inverted orientation, meaning they're pointing at each other rather than being pointing in the same direction as each other. So the first thing we need to do to get this recombination event to occur is to pair those 2 so that they can actually undergo recombination, and just then look at the outcome. Okay, so I'm pairing these for recombination by making this little lollipop shape, where the red arrows represent the repeated sequence, the F8A sequence. So we can follow this by, again, starting at the left, crossing over where the crossover occurs, follow the loop around, and then look to see what the outcome is. And what the outcome is an inversion of the sequence within the loop. Okay, so I'm following this around with little arrows in yellow, and we're not going to lose anything, but we're going to flip over the orientation as a result of this exchange. So the ultimate, after you've done that, is instead of having F8A promoter, exons 1, 2, 3, 4, etc., F8A, we have an inversion where we have F8A, and then we have the exons 4, 3, 2, and 1, then a promoter, then the other F8A, and then the rest of the exons. So this promoter could drive expression of exons 1, 2, 3, and 4, but exons 5, 6, going up to, I forget, maybe 20, that would be lost. So this gene is totally messed up because it's got an inversion within it that was promoted by the repeat recombination. So that's not a good thing to have happen.